let's delve into the realm of medical breakthroughs. The fight against cancer has been a defining challenge of our time. And today, we stand on the brink of a groundbreaking discovery. With the tireless efforts of two brilliant researchers, we have reached a pivotal moment where hope is no longer just a distant glimmer but a tangible reality. Welcome to this extraordinary event where science and innovation collide, bringing hope and excitement for a cancer-free world. Tonight, we present to you the findings of this novel invention of nanoparticles to arrest the human lung carcinoma cells that could revolutionize cancer prevention. We have with us Dr. Joan Jacob, Assistant Professor at the Department of Physics, Marivanius College, Thiruvananthapuram, and Mr. Jyothish II, Assistant Professor at the Department of Physics, Sri Narayana College, Varkala, to share the details of their research.
Using nanotechnology in medical diagnosis and treatment is taking the healthcare sector to higher altitudes.
In nanomedicine, scientists seek nanomaterials with much better curing effects than conventional medicines. The new nanomaterials can sometimes be mixed with the existing drug to create better results.
We could select only those materials which are non-toxic as well as biocompatible. It will not cause any side effects or should have minimum and bearable side effects. Ferrites are such materials. We know that iron is naturally existing and a non-toxic substance. Its oxides are also abundant. Nearly 90% of lung cancer is from direct or indirect use of tobacco or related products. Worldwide cancer research has brought many types of cancers to curable levels, but still lung cancer is a big challenge. In this scenario, we investigated the anti-cancer activity of various nano-sized ferrites, especially against lung cancer. We have experimentally determined the anti-cancer activities of different ferrites and surprisingly got a very outstanding result for a particular combination. In our research lab, we were preparing ferrite samples through chemical methods. Each ferrite sample was prepared by precipitation of the appropriate raw materials. The samples were well purified and were given precise thermal treatment before finalizing the product. We all know nanomaterials have unique properties because of their crystalline size at the nanometer level. We have conducted various investigations to determine our samples' size, morphology, and purity. They include X-ray diffraction and electron micrographs. We have given more importance in investigating the anti-cancer activity of these non-toxic ferrites.
Our research found that silver-doped zinc ferrite nanoparticles work effectively against human lung cancer. It controls the spread of cancer cells. Cancer cells have an abnormal property that they grow and divide at a rapid rate. Our body has a natural mechanism of destroying unwanted cells, and this mechanism is called apoptosis. Genetic engineering has identified that a human gene named p53 has a high potency to induce cell death. Cancer researchers are going in a direction to seek methods to stimulate the gene p53, hence controlling the cancer cell spread. Several researchers got good results by using metallic nanoparticles in cancer treatment.
In our study, we conducted the anti-cancer activity of silver-doped zinc ferrite nanoparticles, specifically on lung cancer tissues. We could observe that our silver-doped zinc ferrite nanoparticles could produce apoptosis by causing the breaking of the cell membrane. Nanoparticles are very small in size, hence they can penetrate through the cell membrane and accumulate within the cells, causing effective destruction of cancer cells. We have estimated the anti-cancer properties of our samples systematically and scientifically. Lethal concentration 50 or LC50 value is a standard measure of the anti-cancer activity of a medicine. It gives the amount of the drug required to kill 50% of cells within a specific period. Large LC50 means that you need a higher concentration of that medicine to cause 50% of cell death. Our studies showed that The LC50 value of our silver-doped zinc ferrite nanoparticles is 7.17 microgram per milliliter on lung cancer cells. This is a trace amount, and it was confirmed that silver-doped zinc ferrite nanoparticles can potentially destroy the lung cancer cells. We have also compared this value with that of the conventional chemotherapy medicine named 5-fluorouracil or 5-FU. This chemotherapy medicine has an LC50 value of 13.73 microgram per milliliter, which is higher than the value of our sample.
When a medicine is administered to a patient, it may kill healthy cells along with the cancer cells. So, we have conducted tests on the destructive effect of our samples on healthy cells. The LC50 value on healthy cells was determined to be 172.9 mg/ml, which is very high in concentration, nearly 25 times higher than that for cancer cells. The result is interesting since we require a deficient amount of the drug for killing lung cancer cells, which does not cause much damage to healthy cells. This is a type of targeted treatment for lung cancer.
The microscopic images showed significant morphological changes in lung cancer cells on treatment with our samples. The cells were detached, shrunken, dispersed, and cytoplasmic shrinkage occurred after treatment with the samples. Our samples can potentially restrict the migration of lung cancer cells and lessen the likelihood of metastasis.
We have conducted several other related investigations to study our samples' effect on lung cancer cells. Some are fluorescence activated cell sorting analysis, migration assay, the sample's impact on the antioxidant systems, indirect ELISA, cell cycle analysis, and finally the mRNA expression of gene P53 and gene P21 by real-time PCR analysis. All these results strengthen our silver-doped zinc ferrite nanoparticles' ability to fight lung cancer. The next step is to test it on animals and then on humans.
You were listening to Dr. Joan Jacob and Mr. Jyothish II, the architects who unveiled the potential of nanoparticles in combating human lung carcinoma cells.